Good afternoon, everyone. First of all, thanks to Professor Ganesh Bagler for giving me this opportunity to present some insights into how into the gut food health axis and how the gut microbiome is modulated by the food that we eat or can modulate the effect of the food that we eat on our health. Okay, so initially when Professor Ganesh gave me the offer, asked me to present. I was thinking about presenting some of my own research, but then I decided not to be too technical, as last time it was a bit too technical, and rather to give some examples of some example research at a very basic level onto this axis. Some example events or anecdotes that have shown different modes by which the microbiome is influenced by the food and affects the health, and at the same time the microbiome can modulate the effect of food on our health. So I see a lot of students over here who may not be aware of the gut microbiome. So I'll just start with a basic introduction to the gut microbiome and the human microbiome in general. So I work on, like Professor Ganesh mentioned, I work on the microbiome. So human microbiome, specifically the gut microbiome. I'm a computational biologist who works on analyzing microbiome data all over the world and identifying its links to various diseases, its association with lifestyle, and looking into all kinds of covariate-based analysis to decipher different kinds of mechanisms undergoing the different kinds of axes within our body. So what is the human microbiome? The human microbiome is basically the huge repository of all different kinds of microbes that resides in various sites of our body. So if you look into the microbiome in terms of pure numbers, so there are around 100 trillion symbiotic microbes that reside in our body. And they belong to various lineages like bacteria, archaea, fungi, viruses, and protists. 95% of these small members of the small entities are actually in our gut. If you look into the number of microbial cells in terms of absolute numbers, they are at least 30% more than what we have as our own cell or what we can call our own. So in a way, we are more microbial than human. However, if you look into the functional genes that they encounter or they encode, the number of genes that they encode in terms of diversity is more than 2 orders of magnitude high. So in terms of the functional potential, they are 100 times more than the functions that we have evolved to do on our own. So in a way, like I said, we are more microbial than human. And these microbes they vary depending on the body site or depending on the site of our body we are discussing about. So oral cavity has very distinct kinds of microbes. Stomach has distinct patterns of microbes. The gut has different kinds of microbes. The skin and the uterus all have different specific patterns in their microbial composition. And each of these microbial communities is adjusted for the specific niche, or they are specialized for the specific niche in which they are residing. Now, in terms of these microbes, in terms of this microbial community, the most crucial among them is our gut microbiome. So the gut microbiome, like I mentioned, it is one of the largest microbial communities in our body. More than 95% of the human microbiome actually resides in the gut. They do a wide range of functions, starting from the nutrient harvest from the food, the optimal nutrient harvest, to regulating or priming our immune system, to preventing colonization of pathogen and making us less susceptible to infections, to actually producing secondary metabolites that do a hell lot of different kinds of functions, right? Starting from, like Professor Ganesh mentioned, regulating the axis between the gut and the brain, that is modulating our behavior, making us less susceptible to different kinds of diseases like Alzheimer's, improving our cognitive function, and at the same time in aspects like regulating our appetite, for example. So this is how key the gut microbiome is. And that is the reason, because it is linked and because it is functionally so diverse and because it is intricately linked to so many different processes in our body, almost 90% of the diseases that have been studied till now have been associated with some changes in the gut microbiome. More mechanistic research into these lines. So this is a figure that was from a paper that was recently published by me and my colleague in the British Medical Journal, which actually shows these links. Okay, so the gut microbiome clearly has 3 different facets. The first facet is it responds to changes that we undergo in our physiology, and they respond accordingly with an appropriate change. The second, that is as a sensor. The second effector is as a modulator. When the gut microbiome undergoes a change, not influenced by body but independent of body because of some other factor like the food and affects our physiology. Some examples of which we'll be giving in the next few slides. And the third is as a translator or as a transducer. Like the gut microbiome is influenced by all these different factors that I mentioned on the right, like genetics, birth mode, geography, lifestyle, gender, age, diet, medications, transmission from our social contacts also. And they are influenced by this, they are modulated by this, and at the same time they modulate the effect of these environmental factors on our health. So let's come into one of these factors, which is the topic of our discussion, which is called the diet. And I'll be giving you some examples into how the diet, gut microbiome and the health axis persists, the different mechanism, and some of the examples in this regard. So the first mechanism by which the first mechanism of this diet gut microbiome axis is, the first mechanism is the diet changes our gut microbiome. So we have different kinds of diet affecting our gut microbiome differently. There are some of the examples I will be giving in the next slides. And this changes in the gut microbiome has an effect on our health. So here is the diet modulates the microbiome and the change in the microbiome influences our health. There is a second mode that occurs in this axis. The second mode is we take the diet, same diet, two different individuals take, but not all individuals respond in a similar manner. And that mode of how an individual is going to respond is actually determined by the microbiome. And we have innumerable, and I'm really happy to, and I'm really proud, I'm really happy that one of our own studies was the first study to actually show this. Where we showed that specific members of the gut microbiome, irrespective of whether they are affected or not, modulate our response to the diet. So all of us, we take the same diet, but some respond in a different manner and some do not respond in the same manner because of the gut microbiome that we have. And there are multiple examples of both these mechanisms. Some of the mechanisms I'm going to summarize here. So the first mechanism is the food modulates the microbiome and the modulated microbiomes affects health. So this is a figure that I really like, and what this figure actually shows is the way our dietary trends have evolved over the time. So initially, and then across lifestyles. So the most basic lifestyle is that of a hunter-gatherer, like the tribes. Then we changed our lifestyle to become agrarian, and then finally the industrialization came through. And in current world, there are different kinds of societies that are still adhering to each of these different lifestyle to a large extent. And if you see into these trends of diet, you see that the hunter-gatherers, they have an extremely low pattern of processed foods, they have a high intake of plant-based as a meat-based diet, and as we go into the agrarian, the plant-based diet start to dominating and the meat-based diet starts going down and the processed foods start increasing. And with the industrialization, what we see the plant-based diets actually going down, the plant source diversity also going down, and industrialized and the animal-based foods and the processed foods going up. You see an exactly same pattern if you look into this is from a large global cross-cohort analysis, and when they looked into the same pattern in the microbiome, in terms of the microbiome function, you see this is reflected in the way the microbiome digests the food. And that actually in the bottom panel, you see that actually is also reflected in the different kinds of diseases. Like if you look into the last panel, you see that the infectious diseases are actually coming down in the later part of the industrialization period, but you see the increase of non-communicable chronic diseases and an increased use of antibiotics. So there is a link between the food and the microbiome that you take, and there have been innumerable studies that have explored these events in mice model, in humans, in different kinds of societies, in westernized societies, in non-westernized societies, in transitional societies, and the common theme of all their findings is like this. So there are specific, and overall this is a generic pattern that have emerged. Like there are specific kinds of food, like fiber-rich food, flavonoid-rich food, seafoods, lentils, whole grain, fermented foods, which always seem to promote a certain kind of bacteria. These bacteria, they're mostly, if you go by the groups, these are bacteria called butyrate-producing firmicutes, Akkermansia, Odoribacter. There are some key bacteria that have always been shown to be influenced by these positive food items positive food items. And this is in turn correlated with optimized nutrient harvest from the food, improved immune regulation, immune homeostasis, improved cognitive function, and reduced cardiometabolic health. That is, reduced risk for diseases like type 2 diabetes, atherosclerosis, NAFLD, things like that. At the same time, other studies, a range of other studies have also shown that higher intake of processed foods like high salt, high saturated fat, red meat is associated with an enrichment of an alternate microbiome. This alternate microbiome, some of the members, known usual suspects are here, Enterobacteriaceae, for example, E. coli is a member of the Enterobacteriaceae clade, Bacteroides, Rubinococcus nevus, which is again one of the inflammation associated species, and the enrichment of these species are in turn associated with impaired nutrient harvest, defective gut barrier. What is a defective gut barrier? It is a porous lining of your intestinal system. When you have defective gut barrier, you tend to have more inflammation in the gut, you have more diseases like colitis, inflammatory bowel diseases, things like that, which in turn damages your lining, and you have other disorders as well as diseases like cardiometabolic diseases, like for example type 2 diabetes, insulin resistance, atherosclerosis, etc. And this is what we have summarized in this in the review that we published in Nature Reviews Gastro. So some of these links are over here. So for example, increased intake of prebiotics, polyphenols, Mediterranean diet is associated with an enrichment of this blue group of taxa over here that you can see. And these are all associated with increased butyrate as well as other acetate as well as some other nutrients that make us less susceptible to diseases like cognitive disorders, inflammation, oxidative stress, cardiovascular disease. At the same time, adherence to these diets negatively promotes some bacteria. Okay, let me just negatively promote some bacteria like Collinsella, Flavonifractor plauti, which actually make us susceptible. So it is the food that modulates different kinds of members of the gut microbiome and increases our susceptibility to different kinds of diseases. One particular example, and what will a hot research topic that is going on, is the effects of processed foods. Over the years, our intake of processed food like different kinds of tins, vegetables to artificial sweeteners, these things have increased. And at the same time, a lot of research is actually going on to look into the harmful effect of these foods. And this is again from a review that I published in Lancet, where they have accumulated different research findings associated with the intake of processed food on our gut microbiome. And as you can clearly see, these processed food, they contain stressors of the intestinal microbiota like food colorants, artificial sweeteners, emulsifiers, nanoparticles. And in a way, what the effect that they have is different, is very varied, and it is completely very detrimental. They have an impact on the microbiota composition, like this Enterobacteriaceae, Bacteroides, Proteobacteria. They go up. They have an effect on the way our intestinal cells, they perform their gene expression, because of which they become weaker, they become much more porous. And at the same time, some of these microbiota that are actually promoted by these foods, they produce something called as lipopolysaccharides, which are themselves inflammation endotoxins or inflammation inducers. And as a result of that, you can see the different kinds of diseases that they make us susceptible to: endotoxemia, chronic inflammation, metabolic dysregulation, and things like that. So this is one of the ways, these are some of the examples by which we have observed the effect of food on the microbiome. Food alters some members of the microbiome, and the alterations of these members of the microbiome associated with impaired health or beneficial health. At the same time, there is a second member. It is not the food detecting everything. There is a second mechanism that we discussed, where the gut microbiome actually acts as a transducer of the signals of a beneficial diet. So healthy food pattern in this case, there are two different observations that we have had in this regard. First is, a healthy food pattern does not have an effect by itself, but it is only mediated with certain members of the gut microbiome. And here is one of the first studies that where we show that just simple adherence to a good dietary regime, that the Mediterranean diet, does not have an effect overall. But only when some individuals have certain bacteria that respond to this Mediterranean dietary regime, they have an effect on improvement of health status. So this was an intervention trial that we did in our previous assignment in Ireland, where we looked into the effect of this Mediterranean diet on a cohort of 600 individuals across 5 different European countries. So half of the individuals were given their own habitual diet, and the other half were basically given a Mediterranean dietary regime. And they were given this diet for like 1 year, and after 1 year we measured the effects. When we measured the effects, what happened was, we did not find any significant association overall between adherence to the diet and improvements of any of the host phenotypes. But on investigating the gut microbiome and performing some deeper investigations, we observed that in a certain subset of individuals, the adherence to the diet, it led to the enrichment of certain good bacteria and certain bad bacteria. And in only those individuals where this pattern was observed, only those individuals had an improvement in their clinical phenotypes, that is reduction of inflammation, retainment of their, because these are all older individuals, retainment of their cognitive function, improvement in their physical abilities and things like that. So again, the bacteria that were promoted or the bacteria that were depleted are the bacteria that have been known in other studies as generic commensals or generic pathobionts of the gut microbiome. Now ours was the first study. There have been a lot of other studies that have shown exactly the same trend, that is adherence of the Mediterranean diet. The beneficial effect is associated with specific bacteria, and these bacteria, when they are high, only then the individuals respond. Similarly, there was another study by the University of Nottingham where they observed the intake of green leafy vegetable was only effective in reducing inflammation or white blood cell profile only when certain members of the microbiome were present, or when certain members of the microbiome were lower. So this is one way where we see that there are some examples of how the bacteria that not only taking the food intake is not only associated with health, but only when there are some bacteria which responds to the food, then you have a response. There are some in the recent 1 year. Okay, now there are some more examples like this. For example, this is a typical. This is another example. Again, this is a figure from the recent article. So again, all of us, we take diet rich in choline or protein rich diet. All of us, we take fat rich diet. Fat rich diet. All of us, we take different kinds of diet. So there are different kinds of diet, like choline rich diet is like a fish, egg rich diet, protein rich diet. However, not all individuals who take the same kind of diet will respond uniformly. For example, if you take a diet rich in choline, not all individuals will respond. In some of the individuals, it will be ineffective, it will have no effect. But in some individuals whose gut microbiome is actually enriched for species like this, the Clostridium asparaginiform, Clostridium citrone, desulfovibrio, Enterobacteriaceae, what will happen is these particular bacteria, they convert the choline in the diet to something called as trimethylamine, and this trimethylamine in the liver is converted to something called as TMAO, or trimethylamine N oxide, which is a proatherogenic metabolite that makes us more susceptible to heart diseases. At the same time, all of us will take protein-rich diet, but not all of us will respond similar to the protein-rich diet. Individuals who have a higher abundance of this particular bacteria, Clostridium sporogens, the protein in the diet, the phenylalanine in the protein in the diet will be converted to something called as PAG. This PAG is another detrimental metabolite, again making us susceptible to disorders like hypertension. Similar examples also exist for fat-rich diet, when certain bacteria like this Clostridium scindens, Lepton bilophila, they produce metabolite, the bile acids like DCA or LCA, which make us susceptible to type 2 diabetes, even cancer. So again, same food results in worse outcomes only for some individuals depending on the resident gut microbes. There is another interesting study and another interesting examples that come up. So some which has come up recently in a study published in Gastroenterology. In this study, what the authors observed was something very startling. So a couple of slides back, I have mentioned that fiber-rich food contains or feeds specific beneficial members of our gut microbiome, which produce something called as short-chain fatty acids and which are beneficial. So high fiber diet, good health. This is what we know. High fiber diet, good bacteria, good health. So in this study, what they did was, the authors, they tried to actually see if we take a high fiber diet and we give it to a set of IBD or inflammatory bowel disease patients, they should have an improvement of health. However, what they observed is, instead of actually reducing inflammation, they started exacerbating it, or the inflammation actually started increasing in these patients. Then they looked into these patients, they looked into their metabolome, fecal metabolome, and what they found out was the fiber that they were giving, it was actually a lot part of it unfermented. And the unfermented fibers are correlated with reduction in the short-chain fatty acid production. Later on, when they investigated the microbiome, they found that the actual responsive bacteria, the short-chain fatty acid producing bacteria, which are the beneficial good bacteria, they were completely absent in individuals. So the same good diet, fiber-rich diet, has a completely different response in certain individuals because they don't have the requisite bacteria that can do the stuff. So in summary, these are some of the examples. I will be very short. This is a 15 minutes log, so I will be very short. So in summary, these examples, again, these are accumulation of multiple studies that have which results which I have shown here, and they show some very startling way in which the gut microbiome interacts with our food and finally with our health. The food modulates the microbiome positively or negatively associated with the host health. However, the gut microbiome can also modulate the way the host responds to the food. Appropriate design of food-based therapeutic strategies need to thus consider the state of the host microbiome as a covariate and modulator. And that's where this immensely opens up the field of AI-driven food plus AI-driven food plus microbiome-based personalized therapeutic predictor platforms. So, like this particular excellent figure from a recent review published by Eran Elinev's lab from Israel shows that previously we had just food, good health. But now, with the advent of AI, with the advent of these multiple covariates, we can actually predict or devise precision nutrition based algorithms which include multiple aspects: microbiome, genetics, disease, with food to improve diet based, precision based nutrition or personalized nutrition therapies. So with that, okay, sorry, the last slide, but there are significant challenges. The significant challenges actually results from, again, like I mentioned, the gut microbiome. The microbiome is hyper variable. There are high interperson variations in the microbiome and the response of the microbiome to diet. So we need better data-driven approaches for microbiome-based host stratification. Secondly, when we are actually trying to understand the effect of the food on the microbiome and on the microbiome of the food, we need to be able to measure the dietary intake of the participant individuals properly. So we need to have improved curation of food frequency questionnaires, including detailed profiling of the constituents. And so for that, these products or the the databases of the COSY lab in IIIT-Delhi, for example, which Professor Ganesh is formulating, will be extremely helpful. Thirdly, we need improved measurement of food by subjects. We have food frequency questionnaires, but we have to be able to measure the intake of the food by the subjects in a very proper manner. So the dietary frequencies, the FFQs have to be collected in a very precise manner. So for that, again, programs like What Dish by Professor Ganesh Lab are going to be very helpful. So with that, I would like to thank the organizers and thank you all for having a patient listen to the talk.